All right, everyone, welcome to our practical session for Zoology Z102. Today, we're going to dive into the parasite Schistosoma, which has a really fascinating and complex life. We're going to focus on its morphology and, importantly, the essential English terminology you'll need to know to describe it accurately.
You know, we put this explainer together right here in our Department of Zoology and Entomology at Mini University. It's designed to help you with your Z102 coursework and really help you master this material for your exams.
So, here's our plan for today. We'll start by figuring out its scientific classification. Then, we'll get a really close look at the adult worms. After that, we'll uncover the key stages of its life cycle, and we'll finish up by reviewing all the key vocabulary to make sure you've got it down.
Okay, let's get started. First things first. Before you even look at a specimen, a good zoologist always needs to understand its systematic position. You know, its exact place in the great big animal kingdom.
Okay, I really need you to commit these two English terms to memory. Schistosoma belongs to the phylum Platyhelminthes. That's Platyhelminthes. These are the flatworms. And within that phylum, it's in the class Trematoda, Trematoda, which is the group we just called the flukes. These classifications are absolutely fundamental.
All right, so now that we know where it fits, let's take a closer look at the adult worms themselves. This is where we'll see the incredible and pretty unique adaptations that make them trematodes.
Now, this slide here gives us a fantastic overview of both the adult male and the adult female worms. Just take a second to look at their general shapes. And you'll notice something really helpful here. The diagrams have labels in both English and Arabic, which should really help you out.
So the first thing you'll probably notice is just how different the two sexes are. We call this phenomenon sexual dimorphism. Look closely. The male is shorter and, as you can see, much stouter, thicker. The female, on the other hand, is much longer and has a very slender, almost delicate-looking body.
But here's where it gets really fascinating. The male has this truly remarkable adaptation. It's a deep channel that runs right along its ventral side, and it's called the gynecophoric groove. That's Gynecophoric groove. Now, this isn't just some random feature. It's basically a cradle. The male holds the slender female inside this groove, often for their entire lives, which makes sure reproduction can happen continuously.
So how do they survive inside the host? I mean, they're in blood vessels with blood flowing all the time. Well, they have a powerful anchor. Actually, they have two. Up at the front end, you've got the oral sucker, which is right around the mouth. And just behind that, you'll see the bigger ventral sucker. These aren't just passive parts. They are powerful muscular organs that are absolutely essential for clinging to the inside of blood vessels against the constant flow of blood.
Okay, so we've looked at the adults. Now let's shift gears and look at the microscopic stages of the schistosoma life cycle. It's really by understanding these stages that we can understand how the disease it causes, schistosomiasis, actually spreads.
This diagram lays out the entire kind of complex journey. Just follow the arrows with me for a second. We see the journey from the eggs that are released from a human host to all these different larval forms that develop inside a snail host, and then finally back to the stage that infects humans again. We're going to focus on the key stages you absolutely have to be able to identify.
First up, the egg. And listen, the most important diagnostic feature of the egg is its spine. It's that simple. If the spine is on the side, we call it a lateral spine. That's characteristic of Schistosoma monsoni. If the spine is right at one end, we call it a terminal spine, and that's what you see with Schistosoma hematobium. This difference is absolutely key for diagnosis.
So what happens when this egg ends up in freshwater? Well, it hatches, and out comes this tiny microscopic larval stage. This little guy is the miracidium. That's miracidium. You'll notice it's covered in cilia. These tiny hairs are beating like crazy, pushing it through the water on a really urgent mission to find and get inside a very specific type of freshwater snail, which is its intermediate host. Okay, after some time developing inside that snail, thousands, literally thousands of this next stage are released. And this is the one you need to watch out for. This is the Cercaria, Cercaria, and it's the stage that infects humans. You can always identify it by its body and its very distinctive forked tail. It's like a little free-swimming hunter that actively looks for human skin and penetrates right through it, straight from the water.
Whew, I know that was a lot of information. So, to wrap things up, let's quickly review the most important English terms from this session. It's really critical that you know these terms by heart. So let's just run through them one more time. Remember, they are platyhelminthes, and more specifically, Trematoda, or flukes. The adults use an oral sucker and a ventral sucker to attach. The male holds the female in a special gynecophoric groove. The eggs are diagnosed by their spine, which is either lateral or terminal. The egg hatches into the ciliated miracidium, and the stage that infects us is the cercaria with its forked tail. These are the terms you've got to know for this topic.
Okay, I'm going to leave you with a question to think about for our next session. Of all the structures we've looked at today, the suckers for hanging on, the groove for reproduction, those specialized larval stages, which one's single feature do you think is the most significant adaptation for this worm's parasitic life? Be ready to defend your answer. We'll talk about it next time. Thank you.